I'm going to present the result of the study that we just finished enrollment and was sponsored by Nico. From all the talks that we have, this morning, we know that, despite all the efforts that we put for, the treatment of the GBM, unfortunately, the outcome hasn't changed in the past 20 years. For many other cancers, this immunotherapy, targeted treatment, they had like a kind of very advantage, like they imagine melanoma 10 years ago compared to now. But unfortunately, it doesn't look like that they work very well for the glioblastoma. In part is because of the heterogeneity of the tumor. As I discussed earlier, we have two types of heterogeneity, temporal and spatial. So for the temporal, as you can see, when you treat the tumor, you treat whatever was responsive to the treatment. So another subgroup of cells, they dominate for the recurrence. And as it was very nicely shown in the previous talk, you're going to get 3 different tumors if you do 3 different surgeries. Other than the temporal heterogeneity, the other one is the spatial heterogeneity. So this is a patient with a GBM. The standard of care for the surgery is removing the enhancing portion of the tumor. So when we do surgery for the GBM, the most part that we remove initially is whatever is in the center, which is the central core of the tumor. So for those of us that, we use intraop MRI, when we get the intraop MRI, there is a little bit enhancement here or there on the surgical wall, and that's the enhancing rim. Most of the time it's not significant, but you go back and you use bipolar and suction and just shave the wall, don't even bother to send it for the pathology, and you get a gross total resection. Then what is left is the non-enhancing leading edge of the tumor. And basically you are treating that with the data that you collected in most part from the central core of the tumor. So the question was, with all of this heterogeneity, are they the same? Are we treating the non-enhancing portion of infiltrative portion of the tumor with the central core? Are they have the same, they are markers? To that end, we designed this study to collect 3 samples, one from the central core, one from the solid enhancing rim, and one from the leading edge, and analyze them from the immunoprofile, from the next-gen sequencing, and the microRNA analysis. To that end, so for all of these patients, we planned 3 trajectories before the surgery, one from the non-enhancing portion, from the enhancing rim and the central core, and collect the tissue separately in, and I show you how it's done, and then send them in an ice-cooled container to the lab immediately, like within 45 minutes, it was in the lab. The goal was to standardize an automated resection for the collection process. Where we, like many other centers, we have the tissue banking, but for the tissue banking, again, like many others, we have to send it to the pathology first. They take whatever they want, and at the end of the day, they send it to the tissue bank. So for this study, we wanted to collect fresh tissue and send the fresh tissue to the lab to have more viable cells. As a matter of fact, we found like a very nice solution for that. They say when you can ask for forgiveness, but you ask for permission. So we just bypass pathology, we collect our samples, send to the lab, because this study was designed for larger GBMs that they had to be greater than 3 centimeters. So we would never have problem with more tissue sending to the pathology. Although they caught us in, I don't know, case 6 or 7, somehow they figured it out, and I got a call from our neuropathologist. So, then we came to an agreement that we keep our stuff until the frozen result is back, then send it to the lab. But still was much faster than going to the lab. So what was different? I know that there are some other studies going on for the heterogeneity. The difference was using the Myriad NICO, we had plenty of tissue. We had like a worth of 1 suction trap from each trajectory, as opposed to doing like a needle biopsy and get just a little bit of tissue. Besides, the way that this APS automatic preservation system works, you from the time that you're collecting, from the time that you're at the suction trap, this next to the suction trap, you can put ice. So you collect the tissue in an ice-cold container, transfer it to another container for transport to the lab. So you keep it as fresh as it could be, for the to be analyzed in the lab. And for all of those, I am going to share with you how we did it, but they are all, the trajectories. We used like a way to, we could specially document where we got it exactly. We have like screenshot from each trajectory, and each of them, they go to the suction trap, we label it, switch it to a new one, go to the trajectory number 2. Then the tissue is being sent to the lab within 45 minutes, and it was always 1 person ready for this, and they would do whatever need to be done, flash frozen or a single suspension. So from the time that we collect the tissue to the time that is processed in the lab, it was something around 45 minutes. Then finally, the tissue is, some of these are done, some of them are in process, flow cytometry, microRNA sequencing, and next-gen sequencing. We enrolled 20 cases of presumed GBM greater than 3 centimeters to this study. And that's the workflow. So it looks a little bit busy, but it's actually, it was very simple. So it's like a group of different people. We had like this group email. When I see a patient in a clinic, first of all, I never delayed a single surgery just for the sake of a study. So I would send an email that this is the patient. We called it NICO study. For NICO study, I'm doing surgery on Friday. See if if it's eligible. Then the lab team, it happened a couple of times that they said that they need something is in back order, so we canceled the case. But if there's no pushback from different centers, then the research nurses, they're gonna check the eligibility. And if everything is ready, we just go with the surgery as it's planned. So the surgery is going to be done in the same day with or without the study. But most of it, we very rarely had a case that we couldn't put the patient in the study because we couldn't get the stuff done before. And at the day of surgery, we just needed like 1 person from the lab to be standby. When we collect the tissue, we page them, they transfer the tissue to the lab and start the processing. And the endpoint was, as I mentioned earlier, to see if there is any difference especially between the non-enhancing leading edge of the tumor in terms of immunoprofile or microRNA sequencing or genetics. I put this picture because it looked good, but basically we don't need NICO Novus for this. We don't need the light, and I actually don't open the dura when I collect the samples. But after the crane is done, then what I used differently, I used a BrainLab cranial set to put it on the knee code, then you can track the tip. So, and I just opened the dura a little bit because sometimes in larger tumors, if you open it completely, you're going to have shift. It's not going to predict where you are. Just like a couple millimeters for the knee code to be able to pass. And then as you can see in this monitor, you can track where you are going. So basically what we do, we go to trajectory 1. When we are in the position, we get a screenshot, we collect the tissue using like some irrigation, we flush the tube, change the suction trap, go to number 2, number 3. Literally, it adds like 10 minutes to the surgery, and you collect like 3 separate samples from the pre-planned trajectories. Since this is a study that navigated Nico, Myriad is my best friend. I mean, I just use it most of the time as a navigated suction, like 70% of the time. You don't have to change the instrument, bipolar and navigated Nico. You just need to put your head up and see in the navigation where you are. You don't need a wand or something. And if it's in a safe area, then I go to the cutting and cut a little bit. This is the case number 1. This is the trajectory that we had from the non-enhancing portion of the tumor, from the enhancing rim and the necrotic central core of the tumor. And immediately after that, we take the tumor out. So even if you have a little bit bleeding from any of these, you don't worry much, because like in 5 minutes, you're right over there. You can just control the bleeding and take it out. Mostly it happens in the enhancing rim. So I typically do it last for the sampling. And these are some other patients that we enrolled in the study. For each of them, we have 3 samples. All of them, we have 3 screenshots. So we exactly know where they are in case down the road, one of them is very different from the other, because we call it central core, but some of them is completely necrotic. Some of them are a mix of necrotic and enhancing. So they are not exactly the same. And finally, this is our last patient that did it. We did it in like April, and we finished our enrollment. So far, we just have the immunoprofiling results, because for the rest of them, for the microRNA and next-gen sequencing, we had to send them outside of the institution. So we wanted to have all 20 cases done. So when we send it to a different lab, at least they use the same batch or the same technique. So it's not going to be different people doing it. But for the flow cytometry, the first goal was success. So more than 80% of the cells, they were viable. So that collecting them in the ice cold container and sending in 45 minutes to the lab was a successful strategy, and it was very easy. It didn't really need a village to do it. Some interesting result is in the non-enhancing portion of the tumor. We realized that we have fewer immune cells, and from different like subtypes, from the MDSCs to T cells to macrophages, compared to enhancing rim and central core. And also the T cells, although there are some differences, like CD8 to CD4 ratio is less in the non-enhancing compared to the enhancing, and the CD4 central memory T cell are less abundant, but basically the phenotype and functional analysis is similar. It's just fewer cells. Generally speaking, 2 to 3-fold less immune cells in non-enhancing versus enhancing, and in some subtypes, 3 to 10-fold less, like MDSCs, T cells, and macrophages. MicroRNA sequencing is in process, but I'm very excited to see what that would show us. And in conclusion, navigation-assisted sampling And semi-automated tissue collection, the way that we did it is very safe and feasible. Literally, it adds 10 minutes to your surgery, and you can collect fresh tissue for analysis in the lab. So far, we learned that there are fewer immune cells in the non-enhancing portion, that the portion that remains for treatment. Maybe that's one of the reasons that immunotherapy is not very helpful after, as you know, combination with the gross total resection. And hopefully with other results that we're going to get, hopefully it's going to change the landscape, the way that we treat this tumor. I would like to thank our team, especially Namit Ahluwalia, that was the PI of the study, and then he left us, so I inherited this. And I'm happy to answer any questions.
Once again, great work. I'm really intrigued by your navigating the myriad. Any pointers or suggestions how to make that successful?
I just found it a great tool. I just use it, even if I don't need to use Myriad, I just use it as navigated suction. It's one hand is bipolar, the other. The only trick is if you're using navigated Myriad as a suction, I probably ask one of my assistants to push the pedal for the bipolar, because it's very difficult to have two pedals, and sometimes you mix them up. But it's very helpful when you are deep in the tumor and then you're not sure that you want to check with navigation. You just need to look at the monitor. You don't have to ask for a wand or anything. So you're still working, just look, okay, good, bad. And, if it's in a safe area away from sulcus or fissures or something, then I switch it sometimes to cutting. If I get bleeding, then switch back to suction and control with bipolar. I found it very useful. I use it for a lot of other surgeons.
I think that's a great technical suggestion. Any questions from the audience? Yes.
Oh, thank you. Sorry. Great talk. So one of the things we did in the past is if you use fluorescence to mark your tumor microenvironment, it kind of helps you localize where you are. So in other words, the tumor bulk fluoresces more vividly than the tumor margin, which is more of a pink color. And that may be something that helps you localize each of those areas in terms of your sampling. The question I have for you, with the APS system, how are you separating out each of those microenvironments when you sample? Yeah. Is it with a different system?
No, it's like 3 different suction traps. So we collect the first one, then switch it, then go to the number 2, then go to the number 3. And when we got all 3 samples, then we put one more just for the pathology, because we are still using the nickel.